que nuestra próxima invitada no la presenté al principio porque ella merece que sea presentada con todos los honores. Así que en la mañana de hoy nos acompaña la profesora que actualmente trabaja en el Departamento de Ciencias de las Plantas de la Universidad de California, Davis. Tiene una investigación con objetivo de descubrir los genes que influyen en la calidad post cosecha de frutas, hortalizas y cereales. Su experiencia abarca desde la biotecnología, la bioquímica y la fisiología molecular hasta el análisis estructural y químico del almidón como biomateria. Ella recibió su bachillerato en biotecnología de la Universidad de Sheffield en Reino Unido con los primeros honores de su clase y su doctorado de la Universidad de Cambridge en Reino Unido, seguido de una formación postdoctoral en Johns Hopkins Center en Reino Unido y en DuPont Agricultural Biotechnology en Estados Unidos. Ella es oriunda de Barbados y se encuentra muy feliz porque puede nadar con las tortugas verdes que viven en esa área de la bahía de Barbados. Así que it's an honor to present the Dr. Diane Beckles.
Gracias. Gracias. Lo siento, pero no hablo español. Sorry. English. So, very happy to be here, and I want to thank all the people who have helped me so far. So, plant biotechnology and food security. So, I want to talk a little bit about the history of food security, what it will look like for the next 50 to 100 years, and how biotechnology can help with this. And then give examples of instances where people have been able to improve crops using new technologies. So for most of human existence, we have hunted and gathered. It's only in the last 125 years of the last 12,000 years have we engaged in agriculture. And only very recently have we applied technology. But as you've just heard, it's going to be increasingly important to use technology to ensure food security. Population has grown astronomically over the last 2,000 years. And there were predictions that we wouldn't have enough food to feed everyone on Earth. But so far, we have been able to keep up by constantly introducing new technologies. And this has to keep growing over the next 100 years because food production must almost double to keep pace. How are we going to do this when the climate is changing and there are challenges with developing new crops? We need crops that are resilient to stress. We need to reduce the loss of crops that happen in the field and after harvest. And we need crops that are more nutritious. And I will argue that it's possible to do this through biotechnology. It is not the only way that we can have resilience, but it is going to be an important way. So, the phenotype or the characteristics, what a plant or a crop looks like, its yield, whether it will survive stress, depends on the environment, but also on its DNA makeup, its genotype. And what we're doing is focusing on that DNA. We know that it's the code that determines traits, and if we can unlock the secret, then we can make changes to DNA and have predicted predictable, we hope, changes in how crops can respond to environmental stress. And so biotechnology we can define as using molecular tools, using our knowledge of DNA and all the new ways of modifying that DNA to create plants that have traits or characteristics that we want. So you may ask, is it really necessary to genetically modify plants? And I say yes. All of the fruits and vegetables and corn and wheat that we eat, that we buy from the supermarket, has already been genetically modified. These crops would not survive in nature because of all the genetic changes that have occurred over 12,000 years. So these are examples of natural wild fruits and vegetables. So this is the original banana. This is eggplant, tomato, apple, kiwi, avocado, carrot, and strawberries. But over the last 10,000 years, we've introduced changes to the DNA to make them what we see in the supermarket. So we have a long tradition of making genetic changes to plants. So a lot of these changes occurred naturally, by mistake, in nature. And they caused changes in DNA sequence, which in turn leads to changes in proteins, which help to determine traits or characteristics. So mistakes like little bits of code inserted, some removed, or some substituted can lead to changes in these sequences that determine traits. So we know enough for many genes what they do, what traits they determine, and we can make specific changes to influence its characteristics. So over the years, we are able to make more refined and specific changes to DNA, and that precision has been very important for plant breeding. So I'm going to talk a little bit about the transgenic technology, which was first developed in the 80s and 90s, and the current way that we have for generating new traits. So with traditional crossbreeding, we take two plants and mate them, and the DNA is randomly shuffled, and then we select the offspring with the best combination of traits from the two parents. This process is very long, and it can take between 10 to 12 years. With GMO crops developed in the 80s and the 90s, you could change one gene. You didn't need to mix two individuals together, but you could transfer a new trait simply by changing one gene. And this is very powerful. And there are many reasons why you may want to do this. So for example, some crops have a long life cycle. Some crops like potato and cassava, manioc, are very hard to breed through crossing. When you make crosses, you introduce negative genes from one parent into the other. And there's some traits like high vitamin A that the gene does not exist in the species or the crop that you're interested in. So for the golden rice, we took a gene from another species and introduced it into rice. So what can we do with the transgenic approach? So this is just an example. First generation transgenic crops, if you have a normal flower that's pink, you could suppress the expression of the gene for color. and make it white. You can overexpress and make it red, or you can introduce a gene with another color and have a completely different flower. So very powerful. And the way we do it, this is the whole process. We determine which gene we want to change. We have to give the instructions on how that gene will be expressed. Then we have to get it inside the plant. Then we figure out which cells have that gene. And then we regenerate a whole plant from the cells that have the new gene. And then we have to test that the plants behave in the way that we want. So to provide the instructions on how that gene will be expressed, we put it into what we called a plasmid vector. So it's just a carrier, and this will contain the information on when and where the gene will be expressed. We then get the plasmid into the plant tissues, and we use a bacteria called agrobacterium. This exists in the soil, and it's naturally occurring. It's able to take DNA and insert it into the plant's genome. And it turns out that all sweet potato tubers, all the sweet potato we eat, contain DNA from this bacteria. So genetic engineering occurs naturally. So in the States, most of the corn and soybean that are grown, more than 80%, contain DNA from bacteria. So BT corn has a gene that kills earworm. So if an earworm eats this transgenic corn, it will die. And the Roundup Ready corn has a bacterial gene that allows it to survive when sprayed with glyphosate. And these traits are very, farmers love them because it allows for high levels of productivity and less loss in the field. So more than 80%, maybe 90% of all corn and soybean are transgenic. So the transgenic manipulation allows you to introduce lots of new traits and characteristics, but it does have that bacterial plasmid as a carrier, and so it's not a very popular way of creating new plants necessary for the future. And so now I introduce you to gene-edited crops. So the beauty of gene editing is that you do introduce DNA into a plant, you change the DNA, but then you get rid of that introduced machinery. And so you have a new plant with a new trait, but no added DNA. Okay? So what we put inside the plant is molecular scissors called Cas9 and a little piece of guide DNA. And so the Cas9 and the guide DNA go into the cell. The guide takes the molecular scissors to that part of the genome that matches the guide sequence, and there's a break. And the cell will repair that break in DNA, and there's often a change in DNA sequence, which can lead to a mutation. So this is a more fancy way of how it happens. So here's our Cas9 with the guide sequence. It matches a part of the genome. It makes a cut, as you can see here. And then that cut is repaired, but the sequence is new, and we can have a new protein with new functions that gives us plants with new traits. So this is going to be very important in developing new crops And we have evidence for this. So we can have small changes in DNA sequence in target genes that can lead to big changes in traits. So the wonderful thing is that it's cheap, it's easy. We have students at UC Davis doing these experiments in the lab. The changes that occur could happen very naturally. We can produce new crops in 4 to 7 years, and these plants are not regulated as GMOs. You can introduce new traits, you can modify complicated traits. It's really good for things like cassava and potato that are hard to breed conventionally. And there are not many problems associated with this technology. So because the USDA has made it easier to get these crops to market, there has been a lot of work to solve some of these problems, to develop new crops resistant to stress, with reduced pre- and post-harvest loss, and with better nutrition and shelf life. So I'll just give you lots of examples so that you can see it's real, that the technology is being translated into actual plants that may be useful. So here we have examples of high producing rice lines. We have examples of tomato that have higher yields, and the bunching makes them suitable for indoor agriculture. By making edits in multiple combinations of genes, you can have synergistic effects. So here, these scientists edited wild tomatoes and produced lines that had high yield, high vitamin A, and bigger fruit sizes. Gene editing allows you to create variation in multiple traits, which is very important. And there are examples where we have resistance to disease, bacterial spec, different bacterial infections in tomato have been reduced, different response to brown streak disease in cassava by modifying or editing 3 genes, better response to disease in cacao, and even in citrus greening. There's some work that shows you can reduce this very severe disease that's inflicting problems in Florida. Also examples of better more nutritious crops in cassava, and in Japan, the company there, they gave away gene editing tomatoes that have high levels of GABA, which is associated with reduced incidence of heart disease. And there's research happening all over the world at a breathtaking pace using this technology to improve crop plants, looking at many traits. It's very exciting. Non-browning mushrooms, a paper came out recently where they have non-browning avocado, better tasting berries and berries that don't bruise easily. And it's not just companies that are embracing this technology. China has invested billions because they see it as an important future for food security for their people. And also Africa. The CGIAR centers are working on crops that matter to them. So yes, banana, plantain, cassava, these crops are being improved, especially to be more robust against disease. And even in my lab, we're working to make a healthier potato using this technology. So I'm going to end my talk here by just saying that, there's a lot of crops that are being modified using gene editing, and some very exciting results are being shown in labs and fields and greenhouses, but we still have to test them in the real world. A lot of genes that are critical to very important agronomic traits have not been discovered. So, we can't use gene editing to improve them yet. The patent landscape is still a little murky. We're not sure how the public will respond. And the European Union is still unsure about, where they land on gene editing crops. But there's just a lot of really exciting things that are developing. Crops that are hard to breed can be edited, so new traits can be introduced quickly. And that applies to not only crops in developing countries, but horticultural crops that were not as economically viable for breeding programs. And now, countries that are resource limited in Africa and Asia are able to take advantage of this technology. We say it's democratized plant breeding. And it's also teaching us how plants work and function by allowing us to genetically dissect or understand really complicated traits. So scientists are excited about how to use it for basic research. And we don't think that the public are going to be as angry with editing crops because we don't have to have that bacterial plasmid in the plants. The plants will have no added DNA. So we don't think that gene editing will supplant or remove the need for traditional breeding, or that it means that we won't need many different approaches for improving agriculture, but we think it's going to make a valuable contribution. Also, really important, and the last thing I'll say is that while advances in genetics and agronomy are important, we have to take into account cultural factors in different countries and regions where we are planning to use this technology. So it's very important to talk to people from different backgrounds and help them to understand how this technology can be used and to have them buy into or be interested in adopting these crops. So that's my talk for today. I hope that you could understand my English. Thank you so much for your attention, and if you have any questions, I would be happy to answer them. Thank you so much. Thank you.
Or in domestic animals?
No.
Can you explain that?
So we've done some work. So it's 30 years in the US that people have been growing Roundup Ready crops and BT crops, and lots of papers have been published. People produce what they call meta-analyses, where they look at all the papers and come up with a conclusion. And so all of those papers say that there's no evidence for any harm to human or animal health. At UC Davis, one of our scientists in the animal sciences department looked at studies of all the beef produced in the cattle industry, because those cows are fed corn that's genetically modified. So the thinking is, if it's harmful to health, you would see some change in animal health, at the same time with the introduction of this genetically modified corn. And they found nothing. So I feel quite confident in saying that these GMOs have had no impact on human health. If you eat too much modified, corn, high fructose corn syrup made from genetically modified plants. Well, that's not good for you. But the evidence is clear. And the National Academy of Sciences, which is the most respected body of scientists in the US and globally, they produced a final paper where they looked at all of the studies, all of the meta-analysis, did their own studies as well, and they felt very comfortable saying that there's no negative effects on human health. So it's very comprehensive, and that's the science. So there are people who will say, well, I ate this and I don't feel very well. That is not science. So that's where the evidence and the data takes us. No harmful effects on humans or animals. Okay. Yes. I need one question.
Según lo que pude entender, si alguien se lo puede traducir, utilizan lo que es el cambio genético en ese fruto o esa planta para modificar y que sea más resistente quizás al cambio climático. Y me voy en la misma línea que el profesor, que el doctor Negrón. No tiene que ver algo de lo que ella explicó con lo que se trabaja aquí en Puerto Rico en las empresas Monsanto.
I got some of it, but I need you to help.
Donde modifican genéticamente las semillas, ¿verdad? Para resistir a diferentes pues, fenómenos, ¿verdad? Que pueden afectar, pero tengo la misma preocupación. Genéticamente eso no puede afectar, por ejemplo, la salud del que consume ese alimento o esa planta.
Oh.
She understood the genetic modification, and she's asking is if these transformations, and you just explained, are similar or related to what companies in the island do with the production of crops or seed. And she mentioned one example, she mentioned Monsanto.
Oh, so you're asking if the methods Monsanto use
If the
or
methods are similar, and also if we are sure then that does not hurt the human.
Okay. So yes, the methods are similar. Whether it hurts human beings depends on how you change the plant. So tomatoes produce a poison in the young fruit. If I change the gene to make that toxic compound be higher levels in the ripe fruit, that's bad for humans, right? But that's the gene, not the technique, right? So we have to make a difference between what we change and how we change it. So the method is okay, but it's how that method is applied. Most scientists want to do good. There may be some people who don't want to do good, but method versus trait. The method is safe, it's used globally, but there are genes that people could change that may be harmful. Those genes would not be, those plants would not be allowed to be produced in the United States. There's some of the other questions. For the gene edited, the biggest cons have nothing to do with the science. Okay, one con is we make a specific change to a gene, but there may be other changes to genes that we didn't intend. But it's easy. We can figure out if that happens, and we wouldn't use that line. We just, choose a line where we have only the change in the gene that we want. Any unintended effects we can discover quickly and not use those plants. So it's a con, really. We can get around it. So apart from that, it's just things like patent issues, and where you can sell the product. So in Europe, they don't want genetically engineered crops. So if I'm a US company producing seeds, I can't, Europe is not going to accept those seeds. Yes, and some companies for a long time didn't want to do gene editing because they didn't want to be associated with it, because it didn't matter if they sold non-edited seeds to Europe. The people there may say, well, you do this, how do you know there was no mix in seeds? So for a long time, people were a little bit scared. And apart from that, it's patent issues. So the University of California and the Broad Institute in MIT were fighting about who owns the technology. Some people had a license with Broad, some with UC, and it was very problematic. And so some companies and countries didn't know what to do, so they didn't engage. So I think that has sorted out now. But Broad Institute owns the technology. They have said for small labs, academic labs, you can use the technology. If you're a bigger company, you have to pay us money, and especially if you're a pharmaceutical company. So some small companies weren't sure, so that was a con. But none of these problems matter for me and for you, or for small scientists or groups wanting to use the technology to improve cassava. Broad doesn't care. For the rich people, yeah. It's rich people's problems. Exactly. Exactly. So it's, in Africa, and everyone is using it. It's really simple to do, and it will democratize plant breeding. And that I am excited about. Usually new technology leaves poorer people behind, people who don't have access. This broadens it. All right. Oh, that is such a good question. I love that question. So what people are discovering is that some of the benefits of the genetic engineering can be transferred when you graft an edited line with a non-edited line, because they're mobile DNA that moves through the plant, and that can transmit some elements that have been changed. But also, if you have a rootstock that is disease resistant and you have the scion, then automatically you have a better plant, because the rootstock can tolerate drought, it can tolerate, non-fertile soil, and so that plant would survive better. So there's two things. There's the environmental aspect, because you may have an edited rootstock that can make the plant more resilient, but there's also mobile genetic elements that sometimes become activated when you do grafting of GMO or edited lines with non-GMO or edited lines. So this is an area people are very excited about this. So that's a really good question. Yeah. Mm-hmm. Yeah, so grapes, walnuts, these kind of things, people are actively looking at using grafting in combination with gene editing for more resilient crops. Yeah.
¿Alguna otra pregunta? Pues le damos las más expresivas gracias a la doctora Diane Beckles por su interesante información. Ella va a estar compartiendo todo el día con nosotros y va a estar trabajando en los laboratorios de la tarde.